This is the influenza presentation and the influenza infectious cycle and why there is no universal vaccine presentation by me, Wasim Musleh.
By me, Jason Paxtor.
Kayla De La O.
And Luis Navarro Vega.
Influenza virus history. The influenza virus was first discovered by Richard Shope in 1933, who was able to isolate the influenza A strain from swine by taking the mucus and lung samples from the infected pigs. The most devastating effect the influenza virus has done is its 1918 pandemic, which was caused by the H1N1 virus with avian origin. This pandemic infected 1/3 or 500 million people worldwide, where 50 million people died as the main cause of this virus. The first vaccine for the flu was developed by Thomas Francis Jr. and Jonas Salk in the 1940s, utilizing chicken eggs, which is still a method to develop these vaccines today. They developed an inactivated influenza vaccine, and it would not be until 1945 that the inactivated vaccine would be used for civilians. Influenza virus characteristics. The influenza virus mainly spread by little droplets, which can come from coughing, talking, sneezing, and when it enters the human body, it is going to infect the respiratory system of its host. Common symptoms of the flu include fever, cough, sore throat, runny or stuffy nose, muscle or body aches, headaches, fatigue, vomiting, and diarrhea. The flu has an R0 range of 0.9 to 2.1, and its R0 value will depend on how infective a seasonal strain may be.
So in this slide, we'll be discussing the statistics over the past decade based on influenza cases, such as the number of deaths, the number of infections, and those who required hospitalization. The influenza virus has had a recent spike in cases in the past decade. A recent increase in influenza cases could be due to numerous factors, but has reported a much higher rate of symptomatic illness, as many will often not require hospitalization. So these numbers right here are estimates based on the years 2017 to 2020, right here specifically, as cases involved in 2017 through 2020 are still being calculated, as most death cases and hospitalization are due to COVID. Death cases can be based off other factors due to the fact that death cases are often low in numbers due to influenza acting more of a booster upon previous cases of infection or illness, and therefore are still being separated from other death cases that do not involve the death of either respiratory cases or influenza cases. The influenza virus more often infects a host rather than ending the host's life. As shown by the data table, the number of cases mostly spike on symptomatic illnesses compared to other categories. This tells us that even though the virus is becoming less lethal, it is greatly increasing in its ability to infect hosts, which would suggest that current forms of preventing the spread of influenza are affecting the number of deaths.
Influenza virus structure. So the influenza has three types of strains. So strain A, B, and C. So strain A and B have 8 negative strands, single-stranded RNA segments, while strain C has 7 negative strand segment. The shape is spherical / filamentous. Strain A is studded with glycoproteins, hemagglutinin, HA, neuraminidase, Na, and M2. Strain B is studded with HA, Na, NB, and BM2, and strain C is studded with hemagglutinin esterase fusion, HEF, and it has a viral envelope. Influenza virus attachment and entry. The influenza virus utilizes sialic acid found on host cells for either HA or HEF for virus C. When attachment is successful, the virus will be endocytized, and the virus can be encoded in 2 different ways. First way is fusion with the membrane creates a pH conformational change in which the RNPs enter through a small pore. And the second way is hydrogen ions being pumped through the ion channel, disrupting protein-protein reactions.
Next, I'll be talking about the influenza virus transcription. This process requires a primer in order to generate a 5' capped 3' polyadenylated mRNA molecule. The RNA polymerase of influenza virus is different from other non-segmented negative sense RNA viruses due to its inability to synthesize and methylate cap structures in order to ensure the virus mRNA contains a cap1 structure at the 5' terminus. RNA polymerase must complete key steps. The first step includes RNA polymerase associating with VRNPS binding to the CTD of RNA polymerase 2 to bring VRNPS closer to the 5' capped RNA. Poly2 CTD interacts with the viral RNA polymerase at several binding sites, which stabilizes the cap binding and endonuclease domain in a transcription-ready conformation. In the second step, PB2 cap bind domain is binded to the 5' cap of the host RNA and PA endonuclease domain, cleaving the mRNA from the 5' cap. In the third and final step, the 3' end of the capped RNA primer is inserted into the active site of the polymerase, where it base pairs with the 3' end of the vRNA template. In continuation with transcription, viral mRNA synthesis is initiated by a cis-acting viral RNA polymerase that is part of the VRNP structure and is bound to the vRNA promoter formed by the terminal 5' and 3' sequence of the vRNA template. Transcription begins after PB2, a subunit of the influenza virus RNA polymerase, binds to the 5' cap structure of host pre-mRNA, and it is followed by endonucleic cleavage of host pre-mRNA. Endonucleic cleavage generates the mature end from the pre-rRNA after the 3' end capped RNA primer is positioned in the PB1 active site in the 3' end of the vRNA. The 3' end of the vRNA template is then copied and rebinded to the polymerase. The elongation process continues along the vRNA template through the active site of PB1. This eventually leads to the polyadenylation by repeating copying of U sequence near the 5' end of the vRNA. The 5' cap of the mRNA is released from the PB2 and binding leads to the recruitment of cellular factors for mRNP assembly. Next, I'll be talking about the influenza virus translation. Viruses aren't capable of initiating mRNA translation because they don't possess the required components. Instead, the virus must use the host cell factors to manipulate transcription for its benefit. Cellular mRNAs possess a 7-methyl guanine structure cap at their 5' end, essential in the recruitment of the ribosome to the mRNA. This process begins by the cap structure being recognized by EIF4E, the cytosolic cap binding protein that together with the 4G and the 4A helicase forms the 4F complex. The cap structure is then bound in the nucleus by the nuclear cap binding complex. In order to translate a given mRNA, the cap binding complex must be replaced by 4E at the 5' terminus. The 4F complex through 4G recruits the 40S ribosomal subunit bound to the F3. The binding of 4G to 4E triggers a conformational change in both polypeptides that enhance the association of 4E with the cap and stabilizes the complex. This configuration of factors are thought to promote migration of ribosome from the 5' end of mRNA for start codon. Next, I'll be talking about viral replication. In the viral replication process of influenza virus, VRNP, there are 2 types of polymerase models, transacting and cis-acting. In the transacting polymerase model, the 3' end of the vRNA template is released from the and P-associated polymerase, allowing it to bind to the transacting RNA polymerase. First step includes the binding of the transacting polymerase to GDTP directed by the penultimate residue in the vRNA 3' terminus and the generation of the triple P APG to be elongated by the transacting polymerase. After the 3' end of the vRNA has been copied, it rebinds the VRNP associated polymerase, while the 5' end of the newly synthesized cRNA prime binds to the transacting polymerase. As elongation continues, the 5' end of the vRNA template needs to be released by the vRNA associated polymerase to allow the transacting polymerase to read through the U sequence near the 5' end of the vRNA to generate a full-length runoff copy of vRNA. After the 5' end of the vRNA has been copied, it rebinds the VRNP associated polymerase, while the 3' end of the newly synthesized cRNA binds the transacting polymerase.
Why is there a new vaccine each year? We have a new vaccine every year because of the high mutation rate the influenza virus undergoes on a yearly basis. This is why we rely on data from countries that are undergoing the flu, as we use their data that they collect about their flu vaccine to help create a vaccine for our flu season in order to make it as effective as possible. Usually these changes in mutations are changes in the RNA strand of the virus, causing for different mutations of the spike proteins that are on the membrane of the virus. Vaccines we use today train our immune system to target these proteins, but because of these proteins having slight changes every year, our antibodies do not bind to these spike proteins with a high enough affinity, making the protection or immunity rate ranging from 20 to 99% on a yearly basis. In addition, because there are mutations every year, the vaccine we make for a specific influenza strain is mainly beneficial for that one seasonal strain, but it still could help in other possible strains that develop down the road. Approaches to making a universal vaccine. Universal vaccines in development are trying to attack the most conserved regions of the virus, which are the hemagglutinin and neuraminidase spike proteins, as well as the M2 ion channel of the virus. Hemagglutinin and neuraminidase are essential spike proteins for entry and release of the virus, and the M2 ion channel is necessary to help regulate the pH of the virus to its surroundings so it does not degrade. Three main strategies: recombinant stock-specific hemagglutinin or neuraminidase, chimeric recombinant hemagglutinin or neuraminidase, and recombinant M2 are approaches scientists are using today to make the universal vaccine. In recombinant stock-specific vaccine, the purpose is to make a vaccine that will allow our immune cells to produce antibodies against the stock of the flu virus, which is more conserved in the head region. However, the problem with this is that in order to access the stock, the globular head region must be removed, and when removing it, this causes a change in the spike protein and makes it difficult for the antibody to find an epitope to bind onto the stock region. In chimeric recombinant hemagglutinin, the vaccine would be developed to try to target the stock region of the spike protein with the globular head intact. But the problem with that is that it would be difficult for our immune cells to produce antibodies for these stock regions and instead favor the head because there's an epitope more accessible for it. Lastly, in recombinant M2, the antibody would target the M2 ion channel, which is a prime target because it is an ion channel, making it less likely to mutate. But the ion channel is much less accessible to antibodies compared to hemagglutinin and neuraminidase. So results so far. The BioNTech pharmaceutical company made a rendition of the chimeric recombinant hemagglutinin vaccine, and it contained conserved epitopes for influenza A and B. It was able to provide some humoral and cellular immunity and broad strain coverage, and it underwent phase 3 clinical trial in Europe from 2018 to 2020, with a total enrollment of approximately 12,500 individuals over the age of 50 years old, as each patient was monitored for one flu season. However, the vaccine failed as the results did not demonstrate a significant difference between the vaccinated group and the placebo groups in terms of illness and severity. Here is our references section. We hope you enjoyed our presentation about the influenza vaccine.